All right, well, maybe we'll get started for the evening. So welcome to everyone. Thank you for making space in your evening to connect with us about biodiversity. So my name is Karen McKendry. I work with Ecology Action Center on wilderness topics, and I'm one of your co-hosts for tonight. And I'll pass it over to Becky in just a second. I do want to say thank you to the other partners tonight. So we have Dr. Erin Cameron, who's joining us from St. Mary's University, and Dr. Jen Frail-Gauthier, who's joining us from Dalhousie University. And we're gathering together online tonight in Mi'kmaq, the traditional unceded and unsurrendered land of the Mi'kmaq people. And so one thing I want to share with you tonight is we will be talking about biodiversity. And I really wanted to have someone here to speak from a Mi'kmaq perspective about all of our Msignogama, all of our relations, all of our human and non-human relations that we have here. Unfortunately, there wasn't fit for that tonight. But something I can challenge you to do, that I challenge myself to do at Ecology Action Center and personally all the time, is to decolonize the way that we think about biodiversity, the other organisms living in Nova Scotia, and how our organizations work on that, and how we connect with our natural resources and the gifts from the land and the water as individuals. We're all treaty people, and there's ways to do better in Nova Scotia. And so let's keep working on that together. And so I'll pass it over to Becky to introduce herself and if she wants to share anything.
Thanks. Yeah, I'm Becky Parker. I work with Nature Nova Scotia. I know there's quite a bit of overlap between EAC's audiences and Nature Nova Scotia, so some of you might already be familiar with me and my work, but we do a little bit of everything in nature. We're a federation of nature groups across the province. And so some of our groups are really into just making natural history information more accessible. Some are bigger on advocacy and speaking up for nature. And we're the umbrella group that represents all of them across the province. So thank you for the acknowledgement, Karen. I actually wanted to make a land back statement, if that was okay. We've made land acknowledgements at Nature Nova Scotia events routinely. And I've just had I guess a work week that made me think we should be stepping up our game over the last week, because we recently released this moose explainer series, and I spent an unfortunate amount of time last week policing the comments on that explainer series, in part because we suggested through that series that government consider giving control over natural resource management within core habitat for the mainland moose to the Mi'kmaq, and really it should have said back to the Mi'kmaq. And those were comments came about as a result of that statement that I had to police. And so that was a reminder for me that even though for those of us who work within conservation or are already within the conservation community, that it's kind of a no-brainer giving that sort of acknowledgement back to the Mi'kmaq, but for some people it is still new and they're not quite on board yet. And so on top of the acknowledgement that we're in unceded territory, I just wanted to add, for clarity from a personal voice, I guess, and a professional one, that we're very much in favor of land back in Mi'kmaq. No harvest plan, dam, or mine should go forward without Mi'kmaq oversight. That's also why I'm really excited to be here tonight, because I think it's kind of, maybe last year when folks followed what was going on with the Biodiversity Act, is something that was really obvious to us at Nature Nova Scotia, was that biodiversity is a really big topic and it confused some people. And I definitely noticed a lack of awareness amongst that same audience that tended to come at us with maybe less than appropriate comments. And so I'm really excited by this idea to share these more niche ecosystems and species and just things that people might not be more familiar with, because there's a connection between knowing about nature, protecting nature, but also acting on those social justice issues that are related to the nature issues we care about. So yeah, thanks for having me here today. And I'm really grateful for our presenters.
Mm-hmm. Yeah, thank you. So I'll share my screen now as we start to dive into the presentation portion. So here's a little bit about what to expect for this evening. Is we are going to attempt that challenging basis of just like, how do we define biodiversity? How do we think about it? What are some of the communities that are obvious to us? And then we're going to transition into our two speakers who are going to share a little bit about communities, biodiversity that you maybe don't think about as much, but you're definitely connected to. So what is biodiversity? How can we conceptualize it? I mean, there are very technical definitions out there, like the variability of living organisms, thought of at the genetic, species and ecosystem level. But I think one of the great visualizations for biodiversity is a spider's web. And I think it's a great fit for a couple of reasons. One, the framing, the idea, the visual comes from nature. So it's better than like thinking about it like a robot or a clock full of gears. So this is a natural image to represent it. Secondly, the web brings together the idea of interconnectedness. So just like when something happens in one portion of the web, it can affect and reverberate somewhere else. So is it with the web of life. Something that happens somewhere can affect other species, ecosystems, individuals connected through the web of life. And the third part is that the web is vital. So for species of spiders that use a web for hunting, they need the web to survive. And that's kind of like us and the web of life and all the other creatures connected to it. It's vital for our survival. So how's the web of life looking in Nova Scotia, in the Mi'kma'ki portion of Nova Scotia? So it's pretty neat. It's actually really impressive considering what a small area that we have to care for. So Nova Scotia makes up about 0.6% of the land we call Canada, but it has 13,604 species. Now, these are species that we know about, that we can name, and also have a rank. So we have some sense of how they're doing. So there's in theory more species out there that we haven't ranked yet, and also ones that we haven't either discovered ever or don't know about yet or haven't learned about yet. So of these species, when I talk about ranks, this is something, a process that the province works on in collaboration with experts. Is each year they're taking a look at where are the, what level of population and status do we have for different species. So they have these different categories called ranks. So, oops. There. The first category to think about are called exotics, and their rank is SNA. About 11% of our species are these. So these are species that have been introduced to Nova Scotia in the last few decades or a couple of centuries. And some of them are exotic and here to stay, and some of them are invasive and causing problems. So one example of this is garlic mustard, one of the most invasive plants in the world. We have this in Nova Scotia and it's spreading. The next rank or another rank is extant or of concern. So this is a really important category. This is where we're keeping tabs on species that are naturally extremely rare or have become rare and are becoming more rare. So this is the basis for then listing a species under, say, an Endangered Species Act later. So one example of an S1 rank species is Atlantic whitefish, which is critically endangered in the world. The next category contains a good 20% of our species, and these, fortunately, are apparently secure to secure. So these would be like your really common species or ones that we have reason to think are a little bit less common but aren't in trouble right now. Interestingly, the vast proportion of our species, about 60%, are called unranked or status undetermined. So these are ones where we either don't have enough information to determine a rank, not enough work has been done yet, but we would like to figure out the rank, or there's controversial information about the rank. So I picked one here, the eastern cougar, so a population of cougar that is a bit controversial across its range. Does it still exist? Are there cougars in Nova Scotia? If so, what population do they belong to? So this is one that's a little bit stuck in the middle of ranks right now. And then sadly, we have our extirpated or historical species. So these are ones mostly that still exist in the world, but we have documented that they're no longer in Nova Scotia. So an example of this is the Atlantic walrus, which still exists as a species, but we know the population in and around Nova Scotia went extinct from the area in the mid-1800s because of overhunting. So these are just some ways to think about the species in Nova Scotia. And now Becky's going to share some highlights about species she's thinking about.
Yeah, so I imagine probably many of you in our audience are hobby naturalists or just into stuff outside. And maybe like me, you find you've become that friend that everyone sends pictures to now, asking, what is this? Or like, can you give me more information on what I'm supposed to do with birds right now? I am that friend for a lot of people. And so I wanted to share with you folks some examples, I guess, of hidden biodiversity that I've been really excited about over the last couple of years. And so one is this iNaturalist sighting from 2020. I think it was Ian Bryson, if I'm remembering correctly, who posted this, but it was a prickly pear sighting. somewhere in Nova Scotia. I think the location is probably blurred because it's obviously a very rare occurrence, as you can tell from this iNaturalist map. Prickly pear is a odd kind of like northern cactus-like plant that I know from my time working in Ontario, but I have never seen in Nova Scotia. And so that occurrence was really interesting. I don't think anyone knows yet the story on this population, if it's a group of native being Canadian, I guess, variety, or if there's something else that's just been planted in a sand dune somewhere in Nova Scotia. But I'm eagerly watching this observation, and I'm keen to see if they survive, because it's an oddity for sure. The next slide is the Atlantic whitefish, which is endangered and endemic to Nova Scotia. Only breeding in a couple of watersheds here on the South Shore. And this photo is by Ian Manning. Researchers in Coastal Action spotted its adults in 2020, I think in the Petite Riviere watershed, after not seeing any for at least 6 years, or maybe a little bit more. So that was really exciting. I had a friend who was working at Coastal Action at the time who got to see some adult fish, and it was a really, really big deal for us. The province is actually currently considering a about 50-hectare forest harvest adjacent to the same lake, so not so excited about that. But yeah, super rare fish that you only find in Nova Scotia, and we recently saw some adults, so really exciting. The next photo is a pitch pine, which is a tree more commonly found in the eastern US and parts of New Brunswick. I don't have a photo of the one that I actually found, which was in the middle of this rocky outcrop between hemlock forests on the edge of the Tobiatic Wilderness area in about what I think was probably 2010, because I stupidly did not bring a camera on this 2-month trip, can you imagine? So I borrowed this one from the internet. And you might be able to make out from this photo that this pine has 3 needles per bunch instead of the 2 or 5 that are more common in our native pines. So this was a weird tree when I saw it, and I knew that right away. There were 3 of them all kind of growing together in this rocky, sandy area, which is pretty typical for this species in its common range, but not so much here. So I went back in 2020 to see if I could find it again. It wasn't too far off this main trail leading into the Tobiatic, but the entire area adjacent to the wilderness area had been clear cut, and so it was gone. Yeah. And my final photo here is just 10 species, all 10 species of Serviceberry that grow in Jabuktuk, some of which are really delicious and all of which are just really pretty. So I've been trying to hone my Serviceberry ID skills because I like eating them. And I also have quite a few near me at home, and they are actually really hard to ID. As you can tell from this photo, they look really similar. And actually some species I suspect or some varieties I suspect are maybe more hybrids than others. But I have a couple in my yard that I'm not entirely sure of because they're kind of fuzzy, but not. Anyway, so kind of a confusing bunch of species, but an example where we have a huge variety and they're super useful and super important too. So these are just, I guess, a couple examples of the biodiversity around me that is a little bit niche that I have had to explain to my friends over drinks or coffee, mostly because of my work, I suppose. I might not have experienced them if I wasn't working in this field or if I wasn't a hobby naturalist. But it's also because of these species that I'm not often surprised by nature anymore when I see things that are kind of odd, because I know now that there's just so much more out there that I haven't learned about yet, or I haven't come in contact with. And that's also why I think learning about niche, or what might seem like niche natural history subjects, is so important for conservation and just for ourselves too. Yeah. So with that, we can introduce our speakers who are going to take us deep into some niche natural history topics. So our first speaker is Erin Cameron, who's an assistant professor in the Department of Environmental Science at St. Mary's. And she works on the effects of global change on soil biodiversity and ecosystem services. So Erin uses field observations, lab experiments, and models in her research, which is mostly conducted in Nova Scotia and Western Canada. I'm really excited to learn about soil biodiversity, because as Erin told us, it comprises a large proportion of terrestrial biodiversity and provides key ecosystem services that are critical for human life, but it's not often considered in conservation actions and management plans. So thank you for being here, Erin, and take it away.
All right, let me get my screen shared. Oh, it's on the wrong. Oh no, embarrassing desktop. Okay. And okay. Yeah, you just see the slides now, right? Okay, good. I changed a few settings that I had forgotten I changed, so that complicated things. So yeah, I'm going to talk to you about soil biodiversity. My talk is not going to be all that specific to Nova Scotia. You'll start to understand why as I'm talking. There's not so much in the scientific literature from here on soil biodiversity. But I'm going to start by talking about what soil biodiversity is, why it's important, and what some of the gaps in our knowledge are. So soil is one of the most diverse habitats on Earth. A gram of soil can contain up to 10 billion individual cells and several thousand species of bacteria. But before we can talk about what soil biodiversity is, it's necessary to understand what soil is. Many people may think of soil as the dirt that gets on their hands or that needs to be cleaned off their vegetables. But the more accurate definition is like the unconsolidated mineral or organic matter on the immediate surface of the earth, which serves as a medium for the growth of land plants and is made up of horizons or layers. So on the left side of this slide, you can see some soil layers. Most soil organisms are living in the upper layers there, the organic material, and also in the topsoil, so the mineral, or like the horizon that combines organic and mineral material. So soil biodiversity includes species that live their whole lives in the soil, but also animals that live part of their lives in the soil. Sorry, I'm just gonna get rid of my picture. Okay. So species that, yeah, some species live just part of their life cycle in the soil or reproduce in the soil, and we still generally consider those to be soil organisms. So you can see some examples here ranging from bacteria and fungi, the little red mushroom at the top there, to Collembola, that's a springtail near the top there, to earthworms, and also larger species like the star-nosed mole, which is in the bottom row there. So that's a species that's actually found in Nova Scotia, as far as I'm aware. One of the ways that we classify soil biodiversity is by size. Since there's just so many species, you have to kind of group them together in some way in order to work with them. So this is just a chart showing the different groups. So typical, analyses often look at like the microflora or microfauna, which are bacteria, fungi, and nematodes, up to rotifers, and then other analyses might look at more the mesofauna, so mites and ticks, the springtails, and then we also, some other studies, or maybe you look at them all together, look at the macro or megafauna, including earthworms and beetles and spiders, for example. So this is one way of classifying soil organisms. Another way is to think about soil food webs. So here's kind of a fake soil food web with some of the major groups. So you have litter transformers like earthworms that do the first steps of litter decomposition by breaking the leaf litter into smaller pieces. And then you have the bacteria and fungi that do the real work of litter decomposition. those can be consumed by protists and nematodes. Similarly, the litter transformers like earthworms can also be consumed by predatory mites. And so, yeah, so there's basically similar to above ground food webs. There's predators kind of at the top, and the difference is, I guess, all the microbes involved, and yeah, a lot of interconnections. So a lot of groups of organisms are feeding at more than one level in the food web here. And you also get some species like plant parasitic nematodes that are feeding on roots too. And a lot of species are feeding on the mycorrhizal fungi that are growing on roots. So it gets pretty complex quickly. But why should we care about all this soil biodiversity? I think it's valuable itself, but it provides a lot of services that we and other organisms rely on. You can see some examples here. So carbon cycling, decomposition, water supply, all of that is critical for human life. And just another similar diagram here. So yeah, just to go into a bit more detail, many drugs and vaccines have come from soil organisms, such as antibiotics, so like penicillin. And soil biodiversity is still considered to have a lot of potential for developing new drugs to deal with increases in antibiotic-resistant microorganisms. Another key ecosystem service provided by soil is storage of carbon. So if we're thinking about climate change, the statistic is apparently that 95% of Canada's terrestrial carbon is stored below ground. So it's really a lot. The soil stores and releases CO2, and organisms within the soil affect the rate at which it enters and leaves the atmosphere. So soil biodiversity is pretty important for carbon storage in the soil as well. So those are just a couple of examples. But despite the importance of soil biodiversity for humans and ecosystems, we still know remarkably little about them. I'm going to show you a picture here to demonstrate this. So the gray is undescribed species and the green is described species. So here the top line are like mammals, fish, some plants, and then moving into invertebrates in the second line. The size of the circle here is proportional to the number of species in each group. So even though we're thinking about mammals all the time, it's really a small number of species, but we have described most of them. Then when we get into the invertebrate groups, like butterflies, moths, there's more undescribed species. So you can see a larger gray proportion in these circles. And now I'm going to show the bottom line, which is soil organisms, just some select groups. Here you can see that the gray or the undescribed species is really the majority of species for all these groups, especially for the nematodes. So yeah, there's just a huge amount of biodiversity below ground, but a lot of it is undescribed. Besides having gaps in what we know about species, there's also huge gaps in what we know spatially about soil biodiversity. This is only what's showing up in the scientific literature, so there is more knowledge overall. But so this is a study we did where we searched for every article we could find that had kind of a larger scale research question. So it had multiple sites looking at soil biodiversity and ecosystem services. You can see that there's maybe 10 or so sites in Nova Scotia as compared to the rest of North America, and Europe have a lot more sites. We're not doing so bad compared to like Russia or something, but it's not great, I guess. So yeah, just to give another example of the same thing, that was one study we did, but this is a reoccurring pattern. So this earthworm study is also one that I'm part of, and Helen Phillips is working here now as well with me. So in this study, we took every paper we could find that had where people had actually gone out and sampled the plot and counted the number of earthworms, even if it was just a few little plots. So there's no published research for Nova Scotia on earthworms even. We know what species are here, but we don't have abundances. And yeah, if you look at a similar study that was done where they took soil samples for fungi, you can see that there's like one site in Newfoundland, I guess, but nothing for Nova Scotia. Bacteria, you can see that they didn't bother to get sites in Canada in general, so maybe that's not a lack of data in Nova Scotia, but yeah, limited spatial data for these, or limited soil biodiversity for these data for these large scale studies. There's also, I guess, looking back at the first data set that I showed, this is globally, so not specific to Nova Scotia, since there's barely any data here. This figure is showing the number of sampling sites that looked at different groups of soil taxa. And what we can see here is that there's a lot of variation in how much information we have about species. So one thing that people might find pretty surprising here is that we globally have more sites where microbes have been sampled than some soil invertebrate groups like soil mites. That's probably because it's easier to do like sequencing and metabarcoding for the microbes than the soil invertebrates. This is just from 2 years ago, 2020. There's already been some new work putting together Columbula data from unpublished studies. So that column's increasing, and hopefully more of these will start to be filled in over time. So we really need to improve on this in order to be able to understand soil biodiversity. But the thing about having limited data for Nova Scotia is that even small studies can give us some new information. So we did this little study on Sable Island invertebrates in 2019. We found, so Sable Island had 573 terrestrial invertebrates. That's including soil invertebrates and like insects, like as well. So that was what was reported before our study. Then we did a 6-day study with these pitfall traps. It's just like a red solo cup with a plastic roof buried in the ground, so the invertebrates fall into the cup. And even though it was only 6 days of research, we found 13 new species that hadn't been identified on Sable Island before. That most of them are species that were found on the mainland, but that's still a pretty, I was pretty surprised. I worked in Finland before this, and I would never have found that many new species at a site. So here's, one example is the common grass ant on the left there, and another is the ornate lady beetle, although it may be a bit more aquatic than a soil organism. We found 5 new species of mites and 6 new species of Columbula as well. So we're trying to work hard to increase our knowledge of soil biodiversity in Canada and globally. And one of the things that we're starting this summer is a soil biodiversity monitoring project. So we have about 24 sets of sites in Canada, and 2 of the sites are in Nova Scotia. So that's exciting. We'll be sampling every taxa that we possibly can at the sites. So we're sequencing for bacteria, fungi, and invertebrates, and then we're also hand sorting the soil and doing wet and dry extractions to get more of the soil invertebrates out. So hopefully there'll be more information on soil biodiversity in Nova Scotia soon. There's also, I think we have more information already on agricultural diversity. I just work more in forests and non-agricultural systems, so I can't speak much to agricultural systems, but in those maps we did include any agricultural data that we could, so you can see that it's really quite limited. So yeah, that's all I had in this talk. Thanks for listening, and if you have questions, you can also email me besides asking in the chat.
Thanks, Erin. We do have a question already about earthworms. I understand that all earthworm species are exotic and maybe invasive, question mark, thanks to repeated glaciation of almost all of Canada. Is this still the generally accepted view?
Yes. So I'll just open my earthworm slide, which was cut shortly before. Oh, that's, I guess it's on the wrong screen now. I'll just, yeah, but so yeah, let me do that again. There we go. So yeah, it's, yeah, as far as we know, there's no native species of earthworms recorded for Nova Scotia. There's at least 15 species. They're probably a bit more now. And yeah, there's these three major groups, species that live in the leaf litter layer, in the mineral soil layer, and deep burrowing. Yeah, and there's potentially some jumping worms that have arrived in New Brunswick recently, from originally from Asia rather than originally from Europe. So we'll see what happens in Nova Scotia, I guess.
And Andre's wondering, how comparable are metrics of biodiversity across major taxonomic ranks?
That's a great question. Jen might have things to say about that as well. So I guess, yeah, so one of the issues is how do you define a species for bacteria or something versus like a soil invertebrate. So there, where when I'm saying species, I'm probably more often talking about operational taxonomic units. So they're different sequences rather than something that has been like plated, that plated and cultured that we can actually see visually as being different. It's just a different sequence. So yeah, there's I guess differences in how you define species across different taxa, which makes it complicated to compare, I'd say. Yeah, I guess I didn't realize that you had been sent my original description that I told Mari about. That was, so I did this study where I tried to compare bacteria, fungi, and soil invertebrates with above ground organisms. And we just treated OTUs as species basically to do it, but there's flaws in an approach like that, obviously.
I'm wondering, I didn't realize how much of a gap there was in Nova Scotia. So what can average citizens, including us nerdy naturalists who have camera phones, does anyone say camera phone anymore? Who have phones with cameras on them. What can we do to improve the knowledge, I guess, especially where so many of these things require genetic analysis to really ID?
Yeah, I think like there is some data that like contributions to iNaturalist are still useful, I would say. They're not, it's difficult for earthworms, which is the main group I work on, because it's very hard to take a good picture of an earthworm. But I think for other groups of invertebrates, it can be quite useful. And we still have tried to use that data for earthworms, just like lots of pictures of things that look like shoelaces. so I think one thing people can do is, yeah, contribute to INAT. Some people do have citizen science projects from time to time. I'm not aware of any real active ones in Nova Scotia right now, but there sometimes are. So that's another way to contribute. Yeah.
And Dusan's wondering about a new organism that was found along the Bluff Trail in the Five Bridge Lakes Wilderness area. Do you know anything about that, Erin? Apparently it was assigned to a whole new phylum. He's wondering if this has been confirmed or has any follow-up been done?
Yeah, I think I know what they're talking about, but I think it was when I was moving to Nova Scotia, and I don't know. It's it's a micro base. Like, I think it was like found by sequencing, if it's what I'm thinking of, but I don't know if anyone else knows. Yeah.
Okay. Well, folks, if you have any more questions, then feel free to stick them in the chat. We'll see if we can get to them later, or Erin put her email up there and you can reach out to her directly too.
I can write it in the chat too, so yeah.
Great, thanks.
Thanks.
I'm gonna go dig in my garden after this.
All right, well, thanks so much, Erin. That's really insightful, but also kind of leaves more questions than answers. But it's exciting realms. It's like a neat frontier. We have no need to sort of colonize other planets when there's so much still to discover and learn about here. Or that's my opinion, I guess. We'll now hear from our second speaker, Jen Frail-Gauthier. And I'll share a little intro about Dr. Jen. So she fell in love with mud at a very young age and knew that marine biology was for her. She was introduced to salt marshes in her first year of undergraduate studies at Dalhousie University and studied them through the end of her PhD in earth sciences. So now she's an instructor at Dalhousie University and has taught courses related to animal diversity, paleontology, evolution, ecology. She also teaches a summer field course, coastal ecology, where she gets to teach students hands-on or knee-deep in the mud. She clearly loves doing science outreach and hopes everyone can be as excited about the mud in salt marshes and all of its cool little critters as she is. So I will pass it over to Jen to share.
It's such a fun intro, Karen. Thank you so much. And maybe, I don't know if my name rings a bell, maybe it doesn't to folks, but my co-instructors like to say that my claim to fame was I found a tetrapod fossil at Blue Beach this summer. So maybe if you saw something on the news, that would be me. And it was on a coastal ecology field trip. So gotta love that you can bring students out and have so much fun and you make crazy scientific discoveries for that. And I love that, Erin, you went first because you kind of introduced some of the things I'm hoping to talk about. And apologies. So in the past, when I've done presentations on Zoom, my mic has decided to like randomly scream, and it happens randomly and it lasts like half a second. I don't know if it'll happen tonight, but just in case you hear a weird sound, I'm just giving you that warning. Maybe it's your wake-up call if it just screams at you. So I'm extremely privileged and honored to be here, actually. I'm a first-generation university student, first-generation scientist, originally from Royal Nova Scotia, close to Annapolis Royal, specifically Tiamu-i-Sipu, which is Clementsport. So I grew up in Nova Scotia and grew up along the Bay of Fundy. So marine biology has definitely been there for me. So I don't know, I can see the chat if things pop up, but does anyone know where this picture's from? Anyone have an idea of where I'm taking this photo? It is Nova Scotia. Okay, true. Yeah, definitely Bay of Fundy. Yep. So along Truro is the same. Yep. And yes, Bay of Fundy. Oh, we're going further southwest, I guess. I don't know. Okay, Cornwallis. in the middle. There's a lot of construction that's happened in this area in the last couple of years. And you've probably driven by it a few times. Yeah, Windsor, yeah. So yes, it is the Bay of Fundy. It's the Avon River on the Minas Basin. This is specifically on the Windsor Causeway. So this is one of the locations I used for my PhD, and it's one of the locations where I take students for coastal ecology. So hence why students are knee deep in the mud. So that's what my presentation is going to be about today. We're going to be talking about the treasures in the mud and really all of these small things. Oh, now I've got the chat open, so it's not going to let me. There we go. Go to the next slide. Or I don't know. There we go. So yeah, my name is Dr. Jen. This is not the Bay of Fundy. This is the other salt marsh that I studied for my PhD, which is Chesapeake Inlet. So you may be familiar with that area. This is on the eastern shore, a much older salt marsh than the Windsor Causeway, and a lot more stinky actually. So it's a little bit more muddy. So again, you can throw things in the chat, but when you think of a salt marsh, what are some of the first things that come to your mind? If you had to define it, if you had to describe it to somebody, like what's a salt marsh to you? Birds, yeah, good one. And I'm so ashamed, I'm not a bird. I'm like, that's a bird, or is that a burb or a borb? I'm not as good in my birds as I am the little invertebrates. The smell, yep. That's one thing that definitely comes to mind. Lost boots, oh, too funny. Tall grass, ticks, insects, biting things. Yeah. So grass and mud, birds, those are like, yeah. And I mean, if I said these are tidal salt marshes that we're looking at, blue carbon. Yes, I had a section on blue carbon. I've kind of modified this from other presentations, but I got rid of it because we're focusing on Meiofauna. But mosquitoes. I went to the salt marsh once to get a photo specifically for something, and I was there for 5 minutes and I had 73 mosquito bites on my leg. Yeah, lots of mosquitoes. So mosquitoes, birds, grasses, yes, lovely Spartina. If this is a tidal salt marsh, of course, what makes that? Because Nova Scotia is actually filled with wetlands, but a tidal salt marsh is something specific. We're dealing with the tides, dealing with the ocean, and we can see that through zonation. So these are coastal areas. And oftentimes, maybe from classes before, maybe from your own naturalist experience, that that zonation between marine to terrestrial is probably like your poster area is going to be the rocky shore, right? So what dictates those differences that we see from marine to terrestrial? Like you can look at that and you actually see zones. What makes those zones? Yep, water. Specific moisture gradient. Yep, water. Yeah, and specifically in this area, of course, it's the tide. So it's the tidal areas that really dictate these communities. And some people may be tide experts and hydrology experts and know that, this is 2 meters above sea level and this is, 0.8 meters above sea level. But even myself, I can see what makes something a low marsh or a low intertidal zone versus a high intertidal zone, just based on the zonation and the organisms that can tolerate these specific areas. Some of them are more marine versus some of them are more terrestrial. And a salt marsh actually has zonations as well. And you can see those zonations not from different types of seaweed like we see in the rocky shore or from layers of barnacles and mussels, but you see it through different levels of grass. So of course, when you think of a salt marsh, you're thinking of it all in the grasses and how important these grasses are. And specific zones have specific tolerances for the organisms that live in this area. And it's hard to, I don't know if these GIFs will show up in my share, but really salt marshes are highly influenced by the tides, and there's specific zones of the marsh that are very definitive based on the tidal height, which hint for the future part of this presentation is really important for monitoring sea level, because your tidal heights, can change through time. And so really, even though the salt marsh is one big entity that we see this big area of mosquito-filled, smelly, muddy grass, there really are specific habitats with specific communities and very specific populations within them. And so it's a lot of grass, but did you know that only approximately, it depends on what you're thinking of this, but really between 1 and 10% of the grass production. So when the grass grows, only like 1% of that or a little bit more is directly consumed by herbivores while it is living. So does anyone know where the rest of all of this primary production from the grass is? Where does it go? Anyone have a idea where all of that grass production is going if it's not being eaten directly while it's still alive? Yeah, going down to decomposition, carbon storage. Oh, but say, like I said, I didn't talk about the importance of salt marshes for our carbon budgets, but really important there. So it's going to decomposition, that's correct. Oh, I always have to minimize this before I click the next slide. It always takes a while on my computer. There we go. So Erin showed a great example of a food web for soil ecosystem. Here's a poster example of a food web that you think of in a salt marsh. This is obviously not Nova Scotia, because as of right now, we don't have blue crab, for example, but we have lots of green crabs in our salt marshes. But when you think of a food web, that's what you're thinking of. But really, if only 1% of the energy is going to these big consumers, and like 90 to 99% is going to the detritus, to somewhere else, like that's a lot of things that need to be fed by the muddy ecosystem, so to speak. And this is where this small food web comes in. So when you're looking, I don't know why that was coming up multiple times. When you're looking at food webs, yes, you have birds, you have fish, you have grass, you have crabs, you have spiders, you have all these macrofauna in a food web. But meiofauna, the little things, aren't included, even though they can reach huge numbers in the salt marsh sediments, and even though they have a huge importance to the breakdown and therefore the recycling of the spartina or the grass energy. And many of you may have never even have heard of some of these organisms before. And like when I said, what is a salt marsh? And people say mosquitoes and birds and fish and crustaceans, and not really thinking about all the little things that are in the salt marsh sediments. And that's where we can find the meiofauna. And so most of the energy goes here, and therefore most of the energy comes from there. So the salt marshes and healthy productive salt marshes that we have, yes, stem on the grasses and the production of grasses, but also depend on a really healthy sediment ecosystem and detrital ecosystem. And so these key players in the salt marsh are the decomposers and what we call detritivores, the things that break down or consume the bacteria that are responsible for the breakdown of the fungi, just the really small things in the salt marsh. And so some examples of those, these are definitely not to scale, because we're looking at organisms here that are probably all less than 2 centimeters in length. Here we have, Erin talked about nematodes. Nematodes are hugely abundant in salt marshes. One little teaspoon scoop of salt marsh sediment can have over 1,000 nematodes in it. So they're very, very abundant. The bottom left image here is actually, again, like a tablespoon scoop of sediment from the Windsor Causeway mudflats. And those are just some of the amphipods, which are like shrimp, and some of the swimming big polychaete worms that feed so many bird species that depend on the salt marsh. So these organisms are really important for breaking down the energy and taking it up into the food web. So why should we use, or why do we care about these little tiny organisms, these Meiofauna? What can they do for us? Why are they important to study rather than just studying the birds of a salt marsh, or rather than studying the grasses of a salt marsh? How do you think using Meiofauna could be useful? You can throw it in the chat if you have an idea. What would make Meiofauna a useful tool to study when you're examining things like salt marsh health or salt marsh ecosystems? Yeah, learning about the health and productivity, climate change, yes. Yeah, bottom of that food chain holds everything up. So if there's something going on down in the detritus, then, you know, we could kind of dismantle our food webs further up. There's even some, think of really some logistical purposes of why studying Meiofauna might be really useful. You know, if you're going to convince the government that you need to study salt marshes, and you can kind of say like, hey, if we do this, whoa, I'm going literally backwards. I'm just like scrolling through my thing. I was trying to scroll up the chat and it like scrolls down. Yes, exactly. Easier to catch than birds. Yeah. And it supports the megafauna. Exactly. So we, you know, our commercial fisheries. See if I can scroll through one more without changing it. There we go. Easier to catch than birds. Oh my goodness, it's such a good example. Okay, I'm going to minimize this so I don't go through it super fast again. I don't know why. There we go. Why it takes forever. So one thing is just kind of the ease of collection, right? You can literally go out with a scoop. Yeah. Go out with a scoop. I've used these tiny little containers. You just put them in the containers, put them in a fridge, you know, easy to put some ethanol on them to store them. So very easy to collect and store. You get high numbers, right? So if you're trying to do bird counts, or if you're looking for macro fauna, you know, you don't necessarily get a lot of numbers if you're specifically looking for mammals that might be using the salt marsh. But with Meiofauna, you can get a lot of numbers, a lot of diversity, and therefore you can get this lots of statistical significance, lots of repeatability of being able to do multiple examinations over a short period of time. Relatively straightforward to examine those. It's relatively low financial investment. I did actually most of my PhD on very little research funding. My supervisors actually didn't have any research grants at the time. So it's quite manageable, I guess, financially anyway. Looking at small things is very time consuming. And what I'm going to talk about is foraminifera. They've been used for decades for other purposes. They're useful. And Meiofauna in salt marsh actually occur in very specific environments. So they can be used as these environmental indicators of, you know, fossils or environmental health or pollution and that sort of thing. And so one specific hidden gem of a salt marsh are the foraminifera. Does anyone know what a foraminifera is? Anyone know? If you heard the word foraminifera before. Maybe not. It is a protist. Yay, Dr. Erin. Yep, it is a protist. Some types of protists look like tiny ghosts. Foraminifera actually look like tiny snails, even though they're protists and not animals. So yes, foraminifera, that was kind of the main focus of my PhD. I looked at other Meiofauna, but foraminifera was my golden child. They're protists, so they're like amoeba that you'd find swimming in pond water. And they make a shell. They're really, really tiny, but not so tiny that you can't identify them. So they can be looked at under the dissecting microscope. Really easy to look at morphological characteristics and therefore determine what species they are. And so even though they're small and they don't move around, they are hugely abundant. So a spoonful of mud could have hundreds of individuals of 3, 4, 5, 6 different species. And because they have the shell on them, they can then fossilize. And so therefore these tiny microfossils can be really powerful in many reasons, not just in a salt marsh. And so forams really are these like little data keepers of any marine environment with over 10,000 species. Some are in the plankton of the ocean, some are only in the muddy sand, some are deep sea, and they can tell us all kinds of things like past sea surface temperatures millions of years ago. So really powerful. But specifically in the salt marsh, these hidden little gems occur in very specific zones of the salt marsh. So I talked about those tidal gradients. Certain species of foraminifera only live in very specific zones. And we know that the zones are very tightly connected with tidal height, therefore sea level. So looking through fossil cores of salt marsh mud, you can actually determine past sea level rise within a centimeter, plus or minus a few centimeters range. They are also really important in talking about pollution because, again, they can be indicators of different types of organic environments. So for things like oil spills and sewage. And so some examples really close to home is that foraminifera were used to monitor past and then present wastewater treatment when the harbor cleanup started in the late 2000s. And that was done with my late supervisor, Dr. Dave Scott. And another PhD student, Saad Mohammed. They can also be used for oil spill monitoring and bioremediation in salt marshes. And a good example, luckily we haven't had big oil spills in Nova Scotia, is they've been used to monitor changes after the BP oil spill in 2010. So really, really important, really treasures in the mud, so to speak. Big things come in small packages. And you can take it back to the big picture. And I wanted to kind of end my discussion about, why do we even care about salt marshes in the first place? Like we've been talking about all the cool things that live in the mud that are tiny, that can tell us about things that are going on in the salt marshes. But why are salt marshes so vital? Why do you love a salt marsh, and why should we keep our salt marshes here in Nova Scotia? Interestingly enough, the Coastal Protection Act, the consultation documents came out, I think today. That was really cool to see. Yeah, climate adaptation, really good one there. Importance of, storms. Think of all the storms that we've had. Salt marshes are a natural buffer to prevent, all those big waves from destroying our infrastructure. Yeah, natural filter, that's a great one. They're really, really useful in filtering. And in a place in Bear River, they're used as kind of a natural sewage treatment plant. Yeah, they're nurseries for commercially important fish and even some birds. They have tasty and important plants, and one tasty sea asparagus or salicornia. Glasswort is quite salty, but it's quite a delicacy, and you can actually find loads of it here at Chesapeake, which is this picture. So yeah, salt marshes are something really important to keep in your mind, just like soil and our terrestrial ecosystems. And I hope everyone can get to go out and explore some salt marshes this summer, maybe on a city nature challenge that we have. But I appreciate everyone listening and popping in the chat. You can email, my email is there. My Twitter handle's there. My Twitter's kind of everything from science to just personal stuff, but it's if you can reach out to me if you have any questions. Like Becky said, I'm always that person that like someone will send something to and be like, Jen, what's this? I'd love to talk about all the cool things in salt marshes.
Yay, thank you, Jen. Your enthusiasm for muddy stuff is so contagious. It does make me want to shed my clothes and go out and get close to all the nematodes. There is a question in the chat. Are there any freshwater foraminifera for now?
Yeah, that's a really good question, because what we call them kind of testate amoeba. So there are these amoeba with a shell. Foraminifera are only brackish or marine, but in the aquatic, like wetlands and ponds, we have something to just called, used to be called thecamebans. Erin, I don't know if you have more what they've been changed to, but testate amoeba is kind of like the freshwater equivalent, but they're the same thing. They just don't have that beautiful coilness on their shell. Just kind of more of a circular shell with their blobby parts coming out.
Okay, got a challenging question there for you. Can you comment on the effects of the construction right on it in the salt marsh at Windsor?
Honestly, I was worried. In 2020, I taught my seaside course virtually because of COVID. We were closed down, and I actually went to Windsor to do virtual sampling and videos for the students. And there was loads of rocks, and the grasses weren't as big as I would normally see in July. But we went this past summer because we were able to be back in person, and sedimentation has kind of increased a little bit further, up from, but the salt marsh is still just as vibrant and muddy. It's not rocky. I don't think there have been any long-term effects. It's in the area that I specifically went to, which is close to where the information center used to be, off at the exit 6 off of the 101. I don't know about maybe other areas close to where they have the actual canal going through, but the marsh is actually still pretty good.
Okay, good to hear from someone who knows the spot well. I have a question for you. So both you and Erin brought up that oligochaetes, worms, are hugely important, probably have a diversity that we don't know very well, and we should get to know better. What would it take to get people to study worms, including nematodes, more? There's like a whole wealth there. Why aren't people diving into that?
I can start by saying nematodes specifically. I love worms. Worm-shaped things exist in every animal phylum. So the worm is the ultimate body shape. But nematodes, even though they're the most abundant animal on Earth, they're really hard to like identify in terms of species. And I don't know, Erin, I see you're nodding there.
Yeah, no, I would say exactly the same thing for that. I don't know how to identify nematodes, and if you try to find someone in Canada who can, there's very few people who work on soil nematodes in Canada, as far as I know. So, that's.
All right, a world of discovery available to the keen mind there. I wonder if things like eDNA and other types of testing that doesn't require the human eye or other tools that we're used to identify whether that will start to help more.
Yeah, I tried to do some stable isotope analysis on nematodes, and I did, but I couldn't separate them by species or anything. So it was really more like what Erin was saying in the soil, like functional groups.
Yeah, that's how nematodes are usually treated for soil as well. Just get the functional group and go with that. But yeah, eDNA could help. I haven't tried for nematodes really. I've tried for with like a standard one is 16S or CO cytochrome oxidase 1, and I've used them for invertebrates in general, but my target wasn't nematodes. So
all right. There's another question here, Jen. Does walking through mudflats or salt marshes cause much environmental damage?
I would say no for places like Windsor. The tidal flushing and sedimentation rate is so high that, one tidal cycle and you wouldn't even know we were there. But going to a place like Chesapeake, it's very low lying. The tide is quite gentle. You can see, if I go on a couple field trips, only a month or so apart, I can still see kind of the path through the grasses where I've walked, but you don't see any disruption in the mud. And really, as long as you're not like disrupting the, uprooting any of the plants, it's not really. And causing little depressions and changes in a salt marsh could open up new areas for new biodiversity and new species to come into that are naturally there. But yeah, I wouldn't encourage anyone to go trampling in a salt marsh anyway, just because, the grasses are important to holding the root system, which holds the sediment, which keeps the salt marsh in place. So.
Okay. Thank you for that. We'll give it just a moment more for any more questions in the chat for either of our speakers. And then Becky will do the closing for the night.
I'm wondering if either Jen or Erin have an idea of what, I guess, species of concern might look like at some point in the future, where there's so much that we don't know currently about these dirty, mucky environments. On the big animal side, at least, or even in the plant world, I guess, we know that a quarter of our plants, fish, mammals, birds, that they're all of concern. So considering the state of the world, there's probably something going on within our mucky environments. But yeah, do we know anything about who's at risk and who's okay yet? I mean, maybe not.
Yeah, I don't. I suppose everything's a functional group, right? Like, at least when I see, I do know in other places in the salt marshes that I study, not so much, but there are other places in North America where different grasses have come in and been invasive species, and that can disrupt, the sedimentation process. And there's places further north that the snow geese will actually kind of destroy all the roots of the Spartina, and which causes erosion of the salt marsh. But that's at the big level, so it's hard to think of anything at the small level on a salt marsh side of things.
Yeah, I guess it's hard at the small level in soil as well. Like, you can identify that, like, destroying habitat in a certain area will like reduce the number of species there. But I guess, like, for red listing or anything like that, you need to know where the species occurs, and then you need some population trends, and we don't usually have that. We've been trying to red list or do the process of red listing for native earthworms in Canada, and in most cases it's like data deficient. We have like a record from 1994 and no one's looked for the worm since then. So yeah.
I do see a comment here. Speaking of biodiversity, I was taught in medical school that humans carry in the order of 3,000 bacterial taxons in our guts. So think about that. There's a lot out there we don't know about. All right, folks. Well, I guess you have contact info for both of our great speakers if you have any more questions or, want to know about worms. It seems like that's a cool topic that we're all interested in. In the meantime, I mean, I would love to have one of these talks again. Any exploration into biodiversity is great. Well, today's actually International Day of Forests, or was it yesterday? I can't remember. We should do something for International Day of Biodiversity, maybe like a recurring, like, niche biodiversity talk. It'd be super fun. In the meantime, if you're looking to engage in citizen science or any other kind of efforts that might contribute to understanding our local biodiversity better, as Jen mentioned, the City Nature Challenge is coming up. So that's one way that you can get involved there. You just need iNaturalist, or you don't even really need iNaturalist. You need a friend that has a camera, and then you can go out, take pictures of stuff and get someone to upload them. And then a whole bunch of people on the back end will be there all weekend IDing them. I know I'll be there for sure. That's a fun way to learn about stuff. Even if you're not like a keen naturalist, you can just put stuff up and someone will ID it for you. There's also always events going on within the Natural History Network. So if you're not already following Nature Nova Scotia or any of our member groups, I mean, definitely join your local Natural History Society. Close to EAC, there's Halifax Field Naturalists who also run events on a regular basis where you can learn about cool stuff. So yeah, check out our website, get involved, and see if we can share some cool links maybe after the presentation, Karen. All right. Well, thanks so much to our speakers for being here and Karen for organizing. Great idea.
Yeah, thank you, Becky, for working on this. Thank you, everyone. And yeah, see you out there.
See you later.